In this video we will discuss the results that shows that galectin-1 overexpression can cause proliferation of the Panc1 cells along with the upregulations of the anti-apoptotic effect in the Panc1 cells. So one thing that we need to understand before proceeding with this results is that what is the experimental setup. So this they have prepared the pancreatic stellate cells with galectin-1 overexpressions and galectin-1 knockdown, and they compare the results of the wild-type pancreatic stellate cells, galectin-1 overexpressed stellate cells, and galectin-1 knockdown stellate cells when they are co-cultured with the Panc1 cells, and the results that they are checked is in the Panc1 cells. This is the experimental setup. So first they did the EDU assay to check the proliferations of these different conditions, and from the images it is clear that the one case where galectin-1 has been knocked down has a less signals for the number of cells, and it is comparable to this wild type galectin-1 cells, but it is still less than the wild type cells that I will show it, as they have also shown the quantitative analysis. But it is clear that for the overexpressions of these galectin-1s, when pancreatic cancer cells are co-cultured with overexpressed galectin-1 PSCs, they see a high proliferations behavior. And the quantifications of this EDU assays is done in the panel F, where we can see that the number of proliferation cells is lowest when the galectin-1 has been knocked down, but for the wild type it is slightly higher, and for the overexpression of the galectin-1 it is further increased, and both of these increased and decreased are statistically significant compared to the wild type. Thereafter they did the transfer assay to check the proliferations capacities, and what they are able to see is that when the galectin-1 has been knocked down, the proliferations capacities of the cells are lower compared to the wild type, and for the overexpressed galectin-1 they can see that pancreatic cancer cells this proliferations capabilities is higher, as it is evident from the images, and these things are OD values of this pancreatic cells are quantified. This in the panel G, which shows the invasion assays that is done in the transwell setup, where we can see that there is an increase in the OD values with conditions going from galectin-1 knockdown, wild type galectin-1, and overexpressed galectin-1 in the pancreatic stellate cells that is co-cultured with the Panc1 cells. And they also did this MTT assays for the 5 days, and they are able to see that is the results of the EDU assays. They did it for the 5 days, and the quantifications shown in the panel E, where they can see that there is an increase in the cell proliferations, where the pancreatic cancer cells are co-cultured with the pancreatic stellate cells that have overexpressed galectin-1, and it is at the middle level for the wild type, and the proliferations are lowest when the galectin-1 has been knocked down in the pancreatic stellate cells. And next they check which part of the cell cycle assays get affected or proliferated, and they are able to see that it's the G2 and S phase mainly gets proliferated compared to the G1 phase when in the conditions where galectin-1 is overexpressed, and in the conditions where galectin-1 is knocked down, we see that S phase and G2 phase has lower values compared to the wild type, and this G2 plus S phase are shown, quantifications of the G2 plus S phase are shown in this panel H, where it is clear that it follows the same trends that is followed for the transfer assay and the proliferations assay. Next they check the apoptotic factors also, but it not factors, they check the apoptotic cells, and what they are able to see that apoptotic cells are high in the case where galectin-1 has been knocked down when they compared with the wild type pancreatic stellate cells, and in the case where galectin-1 is been overexpressed, they see that lower apoptotic cells, and these things are also quantified in the for each quadrants. For one step is shown in the bottom side of the panel D, and thus the multiple repeats are shown in the quantifications of multiple repeats are shown in the panel I, where we can see that percentage of apoptosis is higher when the galectin-1 has been knocked down compared to the wild type, and when the galectin-1 is overexpressed, the percentage of apoptotic cells are low. So from these whole results, they finally conclude that this galectin-1 overexpression has two effects. First, it will cause proliferations of the Pancreatic cells. At the same time, it will have it will cause these upregulations of the anti-apoptotic. It will cause the upregulations of the anti-apoptotic effect that will cause less apoptosis of the cells. That's what they have concluded from these results that are presented in this figure. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.